So we're picking right back up where we left off in class, and we had just talked about many of the methodologies like fMRI, MRI, PET scans, things like that. And what's really important to comment here is that when you're using functional methods like PET scans, like fMRI, functional magnetic resonance imaging, you need to keep in mind that the brain is active all of the time. This whole you only use 10% of your brain idea is just a myth. Your brain is always um, uh, in having excitatory and inhibitory activity. There's always blood flow going through the brain, and anytime there isn't blood flow, then that's an issue, and that will actually lead to cells dying because your neurons need to have oxygen and glucose for energy. And so the functional methodologies like PET and fMRI, they run on the idea that anytime you're using a part of your brain, you need energy to that part of the brain, and so blood flow increases to that part. And of course, when you are doing certain cognitive tasks, then there should be an increase in blood flow to that particular area. Now, the one downside is, of course, that there may be a very, very slight increase to the blood flow, and that might not be readily apparent based off of some of the methodologies. So the idea is that we are specifically looking at change from what we call a controlled task, which is when you're not doing anything, to this experimental task, and then you basically subtract out the activity to isolate what we think is activation for that particular cognitive task. And so one challenge that we have, especially when looking at something like fMRI, is that everybody's brain is a little different. We can find landmarks in most normal, typical developing brains. However, the shape and size do change from person to person. And so you might take one person and you see activity in a particular area and say, well, we would like to correlate this activation area to whatever task. But if you just have one person, we can't be for sure that is in fact something that we can say is across a lot of different people. So the idea is that you get a lot of participants to do an experiment, and then you average out that activity across everybody, and you want to see if you get the same activation in the same parts. Now, this is kind of a schematic of how you would essentially do that. If you have a condition here that's actually something that you're doing in what we would refer to as an experimental condition, they perhaps every time they see particular gridded image, they have to press a button, or they have to think a certain thing or make a decision or whatever it might be. And then if you have a control condition where there's, let's say, a fixation point and they're not doing anything, then this right here, this activity should be basically them sitting still, not doing anything. And then this activity should be the person engaging in whatever experimental method that you have. And so then when you subtract these two, whatever's left over should be what the activity is associated specifically with this experimental task. And so you're going to take this subtraction technique and you're going to do it for each participant that you have. And then you're going to average these and get an average activation across multiple participants. And again, the idea is that you're trying to see what the activation is going to look like when you consider more than one person. And if you get lots of overlap in activation, like for example, right here, this average activation right here, then you can say, well, we can say that when you're doing this particular cognitive task, that your brain is probably more active. There's more blood flow in this particular region of the brain. And when you are using different techniques, there are pros and cons of each one. And so we typically talk about this speed accuracy trade-off. So there are some techniques that have really great what we call spatial resolution. It's very, very detailed structural pictures. They look really, really clear. You can, see them at, for example, 1 millimeter to 3 millimeter slices and get a really good visual on the brain. And so some of these techniques like fMRI are really good spatially because you can really view the brain very clearly. Now, the problem with fMRI is that because you're waiting on the blood flow to happen, you are essentially, there's a delay. And so temporally, so over time, okay, the period of making the images and capturing that moment, those, very, very slight seconds between task beginning and then blood flow and then that brain structure being activated, that temporal or time related information is not great with fMRI. So they have really high spatial resolution, but really poor or low temporal resolution. And so you can't track these dynamic changes in the brain very well with a methodology like fMRI. Spatially, it's fantastic because there's really nice images, but temporally over time, it's really difficult to nail that down. And so, of course, if you look at multiple techniques, we can see that some are better than others. And so when we consider this sort of speed accuracy trade-off, fMRI is having this really high spatial resolution with this great detail, but really poor temporal resolution. A technique like electroencephalography or EEG has really fantastic temporal resolution. It's looking at electrical activity in the brain, and it sort of like real time measures this electrical activity. But spatially, it's not very good. It doesn't, because you have electrodes on the surface of the scalp, it's really difficult to know exactly where in the brain that electrical activity is occurring. So spatially, it's not the best, but temporally, it's fantastic. And so here you can see how things overlap in terms of, so this is like MEG, EEG, this is PET, and then here we have fMRI, and you can kind of see temporally which one can really capture the activation and the activity. And here, EEG is just really fantastic when it comes to temporal resolution, but here it doesn't do well with spatial resolution. Here, fMRI does fantastic. Here, the higher, the lower, it's kind of confusing here, means the better resolution, the higher resolution. And then here, taking, as many as minutes, hours to really get that speed down, really poor temporal resolution. So, what could we do? We can actually combine some of these methods and try to get a better count of the person's brain in terms of spatial and temporal resolution. But these methods can be very expensive. And so it's difficult sometimes to do combinations if you are not in a high resource rich institution, for example, because you might only have access to MRI, fMRI, you might only have access to EEG. And PET scans are very, very expensive to do. And that whole process is very expensive. So you may not be able to combine these measures if you are in an institution that is not highly resourced. And plus, typically scientists will sort of specialize in one methodology over another, but we do have collaborations that can combine them. So really, being able to combine methods is the best way to see both a good temporal and good spatial methodology.